Estava dando online, cachorro latindo, campainha tocando, interfone.
Não, a grande questão é que ele não toca. Posso te dizer que foi a primeira ou segunda vez que ele tocou, mas tudo bem. Deixa eu botar para gravar.
Estava dando online, cachorro latindo, estava dando online, campainha tocando, latindo, interfone, campainha tocando.
Não, a grande questão é que ele não toca. Não, a grande questão é que ele não toca. Posso te dizer que foi a primeira ou segunda vez que ele tocou. Posso te dizer que foi a primeira ou segunda vez que ele tocou, mas tudo bem. Eu vou sair. Não, isso aqui é porque a gente está mostrando. É porque está mostrando.
Campainha tocando, interfone, campainha tocando.
Não, a grande questão é que ele não toca. Não, a grande questão é que ele não toca. Posso te dizer que foi a primeira ou segunda vez que ele tocou. Posso te dizer que foi a primeira ou segunda vez que ele tocou, mas tudo bem. Eu vou sair. Não, isso aqui é porque a gente está mostrando. É porque está mostrando.
Campainha tocando, interfone, campainha tocando.
Não, a grande questão é que ele não toca. Não, a grande questão é que ele não toca. Posso te dizer que foi a primeira ou segunda vez que ele tocou, mas tudo bem. Eu vou sair. Não, isso aqui é porque a gente está mostrando. É porque está mostrando.
Campainha tocando, interfone, campainha tocando.
Não, a grande questão é que ele não toca. Não, a grande questão é que ele não toca. Posso te dizer que foi a primeira.
Bom dia, good morning.
Foi a primeira ou segunda vez que eu queria agradecer a participação de todos neste evento. Esse evento é mostrando. É porque estamos nos 3Rs. E também no estímulo para o uso da metodologia, dos métodos alternativos. Gente, vocês não estão entendendo, o telefone está fora do gancho. Como assim? Eu estou falando sério, o telefone, eu tirei do gancho. Como é que ele está tocando? Só um minuto.
Poltergeist. Ok.
Desculpem, porque essa eu nunca vi na minha vida, tá? Eu vou começar a gravar de novo, porque ficou muito esquisito. Essa foi ótima. Heber, por favor, começa a transmitir, tá? No YouTube, por favor.
Já tá transmitindo já.
Obrigada, Heber. A gravação que está demorando. Eu não vou conseguir ouvir. Eu vou parar também de compartilhar a tela. Então, eu vou começar de novo, já que a gente começou a gravação. Então, primeiro eu queria agradecer a presença e a participação de todos. Esse evento, na verdade, são dois eventos acoplados, o International Meeting, porque esse evento é em conjunto com a pós-graduação em Ciência e Biotecnologia do Instituto de Biologia da Universidade Federal Fluminense, onde a gente se localiza aqui em Niterói, no estado do Rio de Janeiro. E também o segundo workshop da Rede de Biotérios da UF, cujo papel dessa rede, formada por mais de 9 biotérios e além do núcleo de animais de laboratório, é ter atividades educacionais que promovam o uso racional, vamos chamar assim, do animais de laboratório, buscando a diminuição do uso de animais, a substituição e a implementação de métodos alternativos. Eu queria agradecer também a participação da mesa. Eu vou passar a palavra para a doutora Evelise Foltas Chagas, que é a coordenadora da pós-graduação em Ciência e Biotecnologia do Instituto de Biologia. Evelise, muito obrigada. Eu estou aqui representando também a pró-reitora de pós-graduação, de pesquisa, pós-graduação e inovação, a própria aqui da UF. Muito obrigada, Evelise, pelo seu tempo.
Na verdade, eu tenho a agradecer, né, a participação e a organização desse evento, né, super importante aí, em conjunto com a PROPPi e a nossa pós-graduação em Ciência e Biotecnologia. E esse evento, na verdade, tem todo o apoio da PROPPi, né? E, além disso, a gente tem muito a agradecer à FAPERJ, porque nós ganhamos um projeto de apoio, né, para o evento, né, para esse International Meeting, e eu Agradeço a Helena, que está o tempo todo aí à frente da organização, em nome do programa de pós-graduação em Ciências e Biotecnologia. Então, né, na verdade, eu thank you on behalf of the program that I postgraduate in Science and Biotechnology, organized this event, the support from PROPPi, and the support from FAPERJ, where I won the support project. Falei só rapidinho, então vou voltar a palavra e, assim, que tenhamos um ótimo evento.
Muito obrigada, Evelise. Só ressaltando, a professora Evelise está falando inglês, porque como um dos eventos é o International Meeting, a gente mantém o bilinguismo aqui, de alguma forma, até porque a palestra da professora Célia Massado será em português, mas a palestra da doutora Esther Pearl será em inglês. Então, é um evento misto. É o primeiro evento misto, na verdade, do International Meeting do PPPI, né, o Programa de Ciência e Biotecnologia, que é sempre todo em inglês, mas dessa vez vai ser bilíngue, né, Evelise? Muito obrigada, tá? Eu vou passar a palavra agora... Obrigada. Eu vou passar a palavra agora para a doutora Carla Eponina, que está representando aqui o Comitê de Ética de Uso de Animais de Laboratório aqui da UF. Lembrando que a CEUA, né, que é assim que a gente chama, ela tem uma interação direta com o CONSEA e essa relação é exatamente para garantir os aspectos éticos e as boas práticas no uso de animais de laboratório. Professora Carla, a senhora poderia falar agora?
Posso sim.
Muito obrigada.
Bom dia, estou tentando aqui ativar minha câmera. Deixa eu ver. Não está me permitindo. Enfim, não está me permitindo, não sei por quê, que eu estou no celular, aí eu não manejo bem a apresentação online no celular.
Tá tudo bem.
Vou falar primeiro, agradecer a todos,
vai ficar no YouTube,
principalmente a PROPPi, a pós-graduação em Ciência e Biotecnologia do Estudo de Biologia, em oferecer essa oportunidade aos animais. Eu acho que eles são os grandes privilegiados, né, quando a comunidade científica pensa em métodos alternativos ao uso deles na ciência e o tanto que eles já contribuíram, né? Então, a tecnologia vem desde a década de 80, em cima da proposta dos 3Rs, buscando alternativas ao uso deles. Agradecer em nome da CEUA a oportunidade também de estar aqui presente, vendo aspectos éticos, né? Os métodos alternativos são contemplados pelo CONSEA, que é o Conselho Nacional de Experimentação Animal. Existe uma RN específica para métodos alternativos. Obrigada, Helena. Obrigada, professora Evelise, por mais essa oportunidade.
Eu que agradeço, viu, Carla? Essa relação na UF entre as questões, né, do trabalho com o animal, e aí, no caso, a rede de biotérios da UF e a CEUA, busca ser o mais integrado possível, exatamente para resoluções de problema, para a melhoria de todos os aspectos profissionais e de formação dos nossos alunos, e também daquilo que a gente pode promover nacionalmente, que é esse investimento que a gente está fazendo agora e que vai se fortalecer no ano de 2024. Eu queria passar a palavra agora para o doutor Alexandre Zibajev, que é professor titular da Universidade Federal de Roraima e que tem trabalhado integrado com a gente. Essa universidade tem colaborado já há algum tempo com a gente, e a ideia é que ele, no ano que vem, ele venha para o nosso pós-doc para trabalhar exatamente nessa amplificação dessas atividades educacionais em nível regional no país, né? Então, que todas as regiões tenham esse acesso. Esse evento já tem a participação de todas as regiões do país, isso já é um fato bem interessante, ou seja, tem gente interessada, gente que está participando, e a ideia é promover isso de uma forma mais incisiva no ano que vem. Então, doutor Alexander, muito obrigado pela sua participação e a sua contribuição com a gente aqui.
Obrigado, professora Carla. Eu fico muito honrado, né, com o convite de participar dessa mesa. Assim, eu fico bastante feliz e assim, bem impressionado com a ideia da professora Helena Carla de expandir não só para o escopo da UF, né, mas para outras universidades, assim, ter um impacto nacional sobre essa estratégia de uso, né, de métodos alternativos, né, e alternativos ao uso de animais. E eu faço parte, como membro suplente do programa de Ciências da Saúde, que é a nossa pós-graduação lá no Centro de Ciências da Saúde. Eu sou professor da parte básica do curso de Medicina, e nós temos na nossa CEUA representantes da zootecnia, né, de outros cursos, curso de Medicina Veterinária, e mesmo suplente, eu participei de alguns eventos, então, assim, eu já mais ou menos sou familiarizado com o funcionamento da CEUA na nossa instituição. Mas a doutora Helena quer, né, expandir essas ideias, né, do replacement, né, da substituição, do uso de animais quando possível, inclusive com novas tecnologias, da qual ela domina. O grupo todo trabalha com métodos em sílico, mas outros métodos também podem ser utilizados. E eu fiquei bastante contente com esse projeto que ela me propôs para o pós-doc no ano que vem, né, fazendo dentro do programa da PPBI. Eu vou tentar contribuir aqui no que for possível. Na verdade, por isso tudo é bastante novo para mim. Eu estou como aprendiz, tem outros estudantes, né, da Helena, que já estão, né, tocando isso de uma forma mais efetiva, mas eu vou me engajar no projeto, né, somando aqui com o pessoal todo. Obrigado, viu, Helena? Prazer.
Muito obrigada, Alex. O interesse nosso de colocar aqui o professor titular, né, de uma instituição federal é mostrar, primeiro, que o pós-doutoramento está aberto para qualquer um, tá certo? E a UF está totalmente aberta para receber quem quiser, quem estiver interessado. Temos pós-graduações nesse assunto, temos graduação também relacionada a TCCs. Então, existe um espaço na Universidade Federal Fluminense para quem tem interesse nesse assunto. A gente está também tentando organizar com a Faculdade de Medicina Veterinária a questão da residência, que é uma coisa importante para a gente, como espaço, inclusive de formação. A nossa rede de biotérios tem espaço para a formação de pessoas no manuseio de animais de laboratório, e essa é a ideia nossa, de formar pessoas mais capacitadas, visada a criar novos métodos alternativos e que seja de interesse de todos, nacionalmente e internacionalmente, por isso a inserção com o NC3R. E agora, vou passar a palavra para a doutora Célia Machado. Primeiro, Célia, agradecer muito a doutora Célia, ela abriu um espaço aí na agenda, todo um encaixe aqui, já deveria termos começado, mas agradeço muito, Célia. A ideia de trazer a Célia é que ela é uma professora renomada na área, ela ganhou o projeto temático, que aí ela vai conversar um pouco sobre isso, mas principalmente o convite com ela é por causa dos temas que ela trabalha em temas de fronteira, como, por exemplo, a nanotecnologia, onde se discute muito sobre como acontece a atividade dessas nanomoléculas e se essas nanomoléculas são tóxicas. E quando você faz esse tipo de pergunta, indubitavelmente pensa-se no teste em animais. Então, ela, na verdade, é como se ela fosse uma cliente, né, Célia? Mas uma cliente que, graças a Deus, se mostra muito preocupada com isso, muito interessada em ver como isso vai proceder, até porque um método mal feito leva a resultados que não são reais, né? Seja para o bem ou seja para o mal, ou seja, indica que tem uma alta toxicidade quando não tem, né? Ou indica um efeito que não existe. Então, ela tem total interesse que os dados, né, fidedignos e éticos sejam liberados por todos que trabalham nessa área. Por isso que eu trouxe a Célia aqui. Célia, muito obrigada por você ter arrumado um tempinho aí na sua agenda, assim como todos, e passo a palavra a você, tá bom? Seu microfone está fechado.
Pronto. Vocês estão me ouvindo agora?
Vou fechar a câmera, tá? Para só ficar você. Eu vou fechar a minha também.
Tá. Deixa eu só colocar em modo de apresentação. Aqui para retornar a lista aqui de apresentação, tá? Tá dando para ver, Helena? Vocês estão me
Tá sim.
ouvindo?
Tá, estamos sim. Tá sim.
Tá joia.
Ô, Célia, você consegue aumentar só mais um pouquinho? É porque a gente fica vendo dois slides, quer dizer, 1,5.
Tá dando para
Você
ver,
consegue aumentar
Helena?
só mais um pouco? É, porque se você aumentar mais um pouco, ele vai ocupar quase que a tela toda.
Vocês estão
Tá
me
sim.
ouvindo?
Tá, estamos sim. Tá, isso.
tá.
Mas está aparecendo...
Deixa eu ver aqui.
Você deu a apresentação de slide, não foi?
Eu coloquei, vê se...
É engraçado. Ah, então é só aí mesmo. Não, tá certo, tá certo. É por causa da linhazinha. Não, tá certo.
É que eu coloquei em modo de apresentação já.
Não, tá certo, tá certo.
Bom, eu vou tentar ser bem breve, né? Como a professora Helena aqui pediu, até cronometrar meu tempo, eu fiz um pouco... eu queria agradecer, Helena, para vir aqui falar um pouquinho do que a gente faz na área de nanomateriais para a saúde, né? Eu, como ela falou mesmo, eu sou uma cliente aí em potencial desses métodos novos para utilização, para não utilização de animais. E aí eu trouxe aqui alguns exemplos, né, de coisas que a gente vem fazendo, não vai dar para, lógico, ficar detalhando nada, mas é mais assim para fazer um geral, né, do que a gente vem fazendo dentro dessa área de nanomateriais para a saúde, né, que o objetivo principal é ir para a pesquisa translacional, né? A gente faz os desenhos, então ir para a pesquisa translacional, aí falar um pouquinho aqui, né? Coloquei aqui alguns pontos aqui principais, alguns highlights aí que eu queria só destacar desse sistema, sem detalhar todos, e falar um pouquinho sobre essa questão da pesquisa translacional que a gente vem trabalhando, principalmente com a minha inserção dentro da rede de nanosaúde carioca, que é coordenada pelo professor Alexandre Rossi, do CBPF, né, que é o Centro Brasileiro de Pesquisas Físicas. Essa rede está desde 2019, e depois que eu entrei nessa rede, a gente vem, então, tentando transformar todos esses materiais que a gente vem trabalhando aqui, desde quando eu entrei na UF, em 2009, tentando já gerar um produto mesmo para que você possa futuramente comercializar, né? E aí, dentro disso, além desse projeto da Rede Nano Saúde, a gente tem outros projetos, como a professora Helena falou, esse projeto que, inclusive, a professora Helena também, ela faz parte da equipe, né, que é o projeto temático de nanotecnologia aplicada à saúde, que envolve desenvolvimento de moléculas e mesmo um método de detecção e diferenciação de arbovirose. A gente também vem trabalhando na validação de imunoteste para a Covid. Isso é uma coisa que a gente já fez durante a pandemia, e agora a gente está na parte mesmo de testar, que não envolve animais, na verdade, a gente está fazendo os testes já em saliva humana, né? Junto com a professora Isabel, aí do Instituto de Biologia, e também temos os sistemas de óxido de grafeno funcionalizados para tratamento de câncer e leishmaniose. Aqui sim, teria, então, tanto no primeiro projeto quanto nesse último aqui, nesse penúltimo, a gente teria, então, animais para fazer esses estudos, né, na parte já pré-clínica. E na rede de nanosaúde, a gente também tem colaborado lá com os pesquisadores para desenvolver diversos materiais também para regeneração óssea, sistemas para tratamento do câncer, e tudo isso envolve estudos pré-clínicos em animais. Então, só para contextualizar um pouquinho, né, que a gente então tem mais essa visão de trabalhar não só no desenho, que seria a pesquisa básica, mas já fazer uma pesquisa translacional, que no caso, né, para quem não conhece aí o termo, né, seria você então trazer a sua pesquisa básica para colocar um produto na prateleira, né? Falando de uma forma mais simples, botar um produto na prateleira que vai ser útil, né? Então, isso envolve, são 4, na verdade, são 5 etapas, né? Que vai de T0 até T4, e essas 5 etapas, então, a gente vem trabalhando, eu digo que a gente está mais ou menos aqui na etapa T2, indo para a etapa T3. Né? Então, a gente faz a síntese, a caracterização, funcionalização, caracteriza depois os efeitos ali desses compostos em células, depois verifica a eficácia terapêutica, e aí começa a fazer os estudos, na verdade aqui na etapa T2, né? Os estudos pré-clínicos, né, da eficácia terapêutica desses sistemas, e depois a gente quer migrar já para a etapa T3 e T4. Né? E aí eu acho que dentro, esses estudos em animais estariam aqui, né, entre as duas etapas T2 e T3, né? Esses estudos, lógico, tanto pré-clínico como os estudos, os ensaios clínicos, né, tem toda uma regulamentação, e eu acho que essa palestra da professora, ela vai ajudar a esclarecer e vai ajudar a gente a fazer, porque às vezes a gente empaca aí nessas duas etapas, né? A gente vai, chega ali na parte animal, ou mesmo na parte já clínica, a gente se enrola um pouquinho. Bom, então, dentro da nanotecnologia, a gente vem trabalhando com esses materiais que eu listei aqui, né? Então, a gente vem trabalhando com as nanopartículas de ouro para diagnóstico, a gente vem trabalhando com materiais de carbono, a gente vem trabalhando com grafeno, para leishmania e câncer de pele, né? São doenças mais de pele. E aqui embaixo, os óxidos, né? A gente vem trabalhando com óxido de ferro, óxido de sílica porosa e também a hidroxiapatita, que é um biomaterial, né? Que constitui os ossos e os dentes. Isso tudo, então, esses aqui seriam usados mais para sistemas de veiculação de fármacos para tratamento do câncer. Eu vou falar, então, brevemente aí de como que a gente aplica isso, qual que é a ideia, né? A gente não utiliza esses materiais puros, né? O que a gente sempre faz é transferir conceitos que são das ciências biológicas, como sítios de reconhecimento, processos de automontagem, sítios de ligação, efeitos cooperativos, multivalência, e usando também a química supramolecular, que é uma química de interações não covalentes, né? Ligações de hidrogênio, forças de dispersão. A gente utiliza esses conceitos para incorporar compostos nas superfícies desses materiais, de modo, então, que eles passam a ter uma funcionalidade muito específica, né? Então, por exemplo, a gente liga anticorpos, tanto anticorpos do vírus SARS-CoV-2, como anticorpos específicos para a Zika, dengue, chikungunya, na superfície de nanopartículas. Eu não sei se vocês conseguem ver aqui, né? Isso aqui. A gente usa sistemas de auto-organização que formam micelas, vesículas, e em volta dessas micelas e dessas vesículas a gente cresce um material de modo que esse material ele apresente ali uma estrutura hierárquica organizada. A gente também desenvolve sistemas que nós chamamos de nanoválvulas, né? Fazendo alusão aí às válvulas macroscópicas. Então, é um dispositivo em escala nanométrica que ele contém um reservatório que a gente coloca um fármaco, a gente fecha esse reservatório e esse reservatório ele só abre em determinadas condições. Então, sob um estímulo, que pode ser variação de pH ou luz, enfim, ou uma reação química, essa válvula ela abre, libera o conteúdo e isso então vai para o alvo. E no grafeno, a gente coloca compostos que são moléculas, polímeros, enfim, ou mesmo compostos naturais na superfície do grafeno para aumentar a biocompatibilidade dele, coloca fármacos na superfície desse material, e aí a gente tira partido desse grafeno, incidindo uma radiação na região do infravermelho próximo, porque o grafeno ele absorve e faz vibrar as ligações, isso gera um calor e, consequentemente, o fármaco é liberado. Então, a gente tem dentro aí desse triângulo, né, a gente junta essa parte da biologia e vai com a ciência de materiais e incorpora isso tudo nesses sistemas para que eles tenham mais biocompatibilidade e eles tenham funções específicas, né? Rapidamente, então, eu vou falar aqui sobre deixa eu só ficar controlando o tempo aqui, né? Rapidamente eu vou falar aqui da leishmaniose. A gente também desenvolvendo dispositivos aí. A leishmaniose é uma doença negligenciada transmitida pelo mosquito palha. O tratamento é um tratamento, os fármacos que existem, né, são fármacos que causam efeitos adversos muito complicados, né? Então, a glucantina ela causa diversos efeitos adversos, é um fármaco que existe desde a década de 40, que é mais utilizado. O que a gente faz, então? A gente coloca um outro fármaco, que no caso aqui seria um de segunda escolha, que é a anfotericina B, funcionaliza o grafeno e aí incide a radiação no infravermelho próximo, que é uma radiação que já é utilizada em terapia, ou mesmo em fisioterapia. Isso, então, faz com que o calor ajude a liberar o fármaco. Então, esse sistema, eles foram testados já, eles tiveram uma eficiência muito boa, então a próxima etapa do trabalho seria agora contaminar a cauda do camundongo com o protozoário e a gente vai fazer uma formulação, uma pomada para colocar na cauda e aí incidir a radiação e ver se tem uma melhora do efeito ou não. Então, aí já a próxima etapa envolveria o estudo animal. Esse outro dispositivo também com grafeno, a gente fez uma funcionalização, colocou um fármaco anticâncer, marcou com tecnécio, né, que é para fazer uma marcação, uma radiomarcação nele, e fez um estudo de biodistribuição em animal. Então, esse aqui a gente já chegou a fazer um estudo de biodistribuição desse material em animal. E a gente viu que esse material ele passa pela barreira hematocefálica, né, cerca de 10%, o que não é muito comum, né? Então, a ideia agora é a gente já incorporou o fármaco para tratamento de doenças neurológicas, a gente colocou taurina e outros fármacos que seria específico, seria um alvo ali para doenças neurológicas. Então, aqui também um outro sistema interessante para estudo animal, e aí estou muito interessada em ouvir, né, como que a gente pode substituir isso. Em relação ao SARS-CoV-2, a gente fez, então, essas nanopartículas de ouro funcionalizadas com diferentes anticorpos aqui do vírus. Fizemos a detecção utilizando uma técnica que é espalhamento de luz. Na presença do vírus, a partícula funcionalizada com anticorpo específico, na presença do vírus, ela se junta, agrega, aumenta de tamanho e a gente monitora esse aumento de tamanho. Então, isso seria o nosso positivo, que seria o controle, e isso, então, nos mostraria, né, que o resultado funcionou. O que a gente vem fazendo, aqui a gente não fez estudos em animais, que é um método diagnóstico, a gente, então, testou primeiro para a proteína S, agora a gente vem testando com vírus na saliva e tanto também no material muconasal, né, para ver e comparando com os dados de PCR. Isso aqui já são resultados com vírus, o vírus, né? Aqui também, que é já o projeto temático, a gente também vem fazendo a detecção, a diferenciação também de arbovírus, a gente quer diferenciar dengue de zika, porque não existe um diagnóstico para esses, né? O diagnóstico são os sintomas, né? Então, a gente não tem um teste para falar, a pessoa está com dengue, está com chikungunya. Então, a ideia seria desenvolver um teste exatamente para isso. A gente fez já os primeiros ensaios na presença do Zika vírus e obtivemos aqui um deslocamento bem significativo, e quando a gente utiliza esse mesmo material, que são as nanopartículas de ouro funcionalizadas com anticorpos específicos para Zika, quando a gente coloca o vírus da dengue, ele não desloca. Então, isso mostra aí que a gente está no caminho, né, para fazer a diferenciação. Finalmente, esse último trabalho, né, são os trabalhos desses sistemas que a gente chama de nanoválvulas, são esses reservatórios que contêm esses fármacos e que eles abrem em pH mais ácido, né? Então, a gente coloca como se fosse uma tampa, né? Então, esse aqui é um dispositivo, dentro dele a gente carrega com o fármaco, que seriam esses quadradinhos rosa. A superfície desse material poroso a gente funcionaliza com um material que se solta dessa superfície quando o pH é ácido e libera o conteúdo. Esse sim também envolve estudos em animais, né? Nós publicamos, enfim, a gente fez todo um trabalho aí mais in vitro, né? E agora a próxima etapa seria já uns estudos em vivo, né? Então, aqui só mostrando, né, que eles apresentaram o material sozinho não é citotóxico para células de câncer de mama, mas quando ele está carregado, ele tem uma liberação lenta e ele vai eliminando essas células, né, com o tempo. Aqui só mostrando, então, as microscopias confocal. Esse aqui seria um dos materiais que apresentou um efeito melhor. Então, em 48 horas, ele consegue eliminar uma quantidade maior até de células do que a doxorubicina pura, que é o fármaco utilizado para esse, né, que a gente utilizou o fármaco de referência. E aí, então, a próxima etapa do trabalho já seria fazer os estudos em animais. Um outro sistema também que a gente acabou de ter o artigo aceito, que seria a gente colocou um biomaterial, que é a hidroxiapatita, a gente sintetizou esse material em 2 metodologias, aqui seria letra A, uma metodologia, letra B, uma segunda metodologia, e esse biomaterial a gente revestiu a superfície dele com essa sílica porosa. A hidroxiapatita, então, ela é utilizada para fazer regeneração óssea, ela aumenta a osteocondutividade, né? Ela aumenta a regeneração ali, mas a ideia é utilizar esse sistema todo para tratamento de câncer ósseo, onde a hidroxiapatita teria a função de recuperar ali o osso, enquanto que a gente revestiu com a sílica porosa e incorporou um fármaco anticâncer, e, ao mesmo tempo, ele trataria o tumor, fazendo essa regeneração óssea, né? Então, esses estudos foram testados primeiro in vitro, a gente viu que também ele tem um excelente efeito, né, para eliminar células. No caso, a gente primeiro estudou células de câncer de esôfago, agora a gente vai passar para células mais relacionadas a câncer ósseo, né? E fizemos também aqui, já a gente conseguiu fazer um modelo, antes de ir para em vivo, a gente usou essas células tridimensionais e viu que esse material ele consegue desestruturar essas células tridimensionais. O grupo que eu faço parte lá no INCA, né, que é onde a gente faz esses testes, eles também são um pouco resistentes em fazer estudos animais, então eles gostam de explorar bastante o que tem ali de ferramenta para fazer os estudos em vivo. Então, esse aqui foi a gente nunca tinha trabalhado com essas células tridimensionais, né, que imita bem um tumor. Então, esse grupo ele tem também essa preocupação de não utilizar animais, né? Mas a gente ainda vai fazer mais alguns testes, mas certamente as próximas etapas seriam já partir para, né, para estudos em animais. Bom, então é isso que eu tenho para falar. Aqui são só mais um resultado que a gente mostra, que foi o estudo que é Living Dead, né, que a gente faz para ver se a célula foi eliminada mesmo ou não, e também mostrou um resultado bastante promissor. E é isso, então, que eu tinha para fazer, para apresentar, para mostrar que a gente tem muita demanda, né, para novos experimentos alternativos aí, que não envolvam os animais. A gente sempre chega na parte de usar um animal, a gente sabe que é uma dificuldade, mas a gente sabe que para avançar para aquelas etapas da pesquisa translacional e para a etapa 4 e 5, a gente teria que ter algo aí nesse sentido. Então, a gente vê que esses sistemas todos, como eu coloquei aqui como final remarks, são sistemas que são extremamente promissores, né? E que isso nos tornaria, então, aí, potenciais consumidores aí dessas tecnologias que serão apresentadas. Então, eu queria agradecer meu grupo, meus alunos, os colaboradores todos, e principalmente as agências de fomento, a FAPERJ, que tem sido uma grande parceira nossa aí, nos financiando, né, com os recursos para a gente conduzir essas pesquisas.
Olha,
Aqui,
muito obrigada, Célia.
a todos. Desculpa se eu
a
passei
gente
um
que
pouquinho
agradece.
do tempo.
Não, tá tudo certinho. Até a doutora Esther já entrou, então tá tudo no tempo, assim, cronometrado. Eu agradeço muito, Célia, até porque eu sei que você tem um compromisso. Eu vou abrir para pergunta, só vou deixar uma pergunta, porque, né, só para a gente cumprir. Existe uma pergunta também no chat que foi feita pelo Leonardo. Eu vou fazer o seguinte, eu vou dar a palavra ao Alexander, e aí a do Leonardo eu vou encaminhar para você, tá, Célia? Para você responder. Eu vou deixar a do Alexander, já que ele vai falar. Oi, Alexander, pode falar, por favor.
Sim.
Doutor Alexander, que é titular lá da Universidade Federal de Roraima.
Parabéns, olha, impressionante, é uma coisa assim que a gente vê que essas ferramentas abrangem, né, um potencial enorme. Eu fiquei bastante interessado pela parte da leishmaniose, né, foi o meu assunto de doutorado, inclusive, eu fui contratado como professor de parasitologia no curso, foi meu curso aqui na Fiocruz, e você falou sobre a compartimentalização dos fármacos, né, na utilização da substituição do glucatímio, que realmente é muito tóxico, a anfotericina, né, ela é menos tóxica, mas também é tóxica. Então, tudo que é tóxico para o protozoário é tóxico para a gente também, porque os protozoários são zoários, né? Nós somos metazoários. E aí é diferente um pouco de bactéria e vírus, né? Que aí são mais específicos, né? E o que acontece é o seguinte, nós temos duas formas de leishmaniose, né? Nós temos a tegumentar, né? E aí o compartimento seria mais o tegumento, né, a forma mucocutânea, né? Mas também temos a visceral. A visceral, inclusive, é pior, né, porque ataca crianças. Então, eu queria saber se você tem feito esse tipo de diferenciação na compartimentalização, né? Quais os que têm chegado, né, na leishmania, se for a tegumentar, se vocês têm tratado também, né, a parte, né, de hepato, nefro, desenvolvimento da leishmania, né, que é a parte visceral, e o glutantime normalmente é de primeira escolha, né? A fotericina é de segunda, né?
Isso.
Então, se tentou também com o glutantime, porque apesar de tóxico, pode ser que ele compartimentalizado seja menos tóxico, tem um efeito menos sistêmico. Então, essa seria a minha pergunta para você, Célia.
Então, a gente acabou escolhendo, sempre quando a gente desenha esses dispositivos, a gente acaba escolhendo, no caso, a gente escolheu a anfotericina B, apesar dela ser segunda escolha, né? Porque para a gente poder fazer uma prova de conceito, que seria monitorar essa liberação desse fármaco quando a gente coloca a radiação no infravermelho. Então, ela absorve na região do VVIS, então seria mais fácil para a gente primeiro monitorar ela, ver se ela está saindo ou não. Como o glucantime ele não absorve na região do VVIS, seria mais difícil a gente monitorar. Então, teria que pensar numa outra técnica para estudar a liberação dele. E talvez, eu não sei se por HPLC, na época a gente não tinha o acesso, agora a gente está com acesso, né? Mas é uma coisa a ser considerada. A gente pensou primeiro na leishmaniose cutânea, porque a gente não sabia como é que seria injetar esse grafeno na pessoa, como é que ele se comportaria. Só que agora a gente começou já fazendo alguns estudos em animais, né? E viu que a gente foi aumentando as doses, né? Até levar o animal ao óbito, e viu que a quantidade para levar um animal ao óbito que você tem que injetar na pessoa, no animal, né, no caso, é muito elevada, muito elevada. Então, assim, já abriu ali uma porta para a gente começar a pensar, por exemplo, no tratamento da leishmaniose já visceral, né? E aí sim, incorporando o glucantime. Seria, então, uma outra ideia que você acabou de dar aí, né? Mas é uma possibilidade também, que a gente achou que na pele seria mais fácil. Aí eu vi a pergunta aqui do chat. De fato, Leonardo, a quantidade está muito alta, a gente está tentando fazer essa validação. Primeiro, a gente está colocando o anticorpo, o vírus na saliva artificial. A gente está com uma dificuldade de fazer na saliva direto, porque dá interferência. De fato, a quantidade é muito alta, e você tem toda a razão. E isso é um dos gargalos aí desse teste que a gente vem usando, né, para tentar fazer a validação, né? Então, assim, a gente está agora tentando ver como que a gente vai fazer para tentar detectar uma quantidade menor. Isso, de fato, a gente já sabia, mas, enfim, a gente está tentando, né, tentando fazer os experimentos, a gente ainda não tem nada... Estamos seguindo aquela RDC da Anvisa para ver se a gente consegue chegar nos limites de detecção menores, e isso, de fato, é um problema, que você apontou muito bem aí.
Obrigado,
Muito
Célia.
Obrigada, viu, Célia? Essa questão com relação à doutora Esther, ela já está até na sala, a doutora Esther Pearl, é que ela vai estar tratando da fase que eu acho que é mais importante, que é a fase antes, né? Que é exatamente o protocolo. O desenho do protocolo ele é essencial para que se consiga ter resultados e principalmente que tenha uma previsibilidade nessas questões, inclusive, de quanto que se usa, de qual é a resposta que a gente está tendo. Então, ter um software que é gratuito, né, e que possa orientar nessa formação no desenvolvimento desses protocolos mais racionais, vamos chamar aqui de econômicos e mais éticos. A grande questão aqui é que muitas vezes a gente dialoga falando sobre medicamentos, e eu posso falar porque eu sou farmacêutica, como se a gente estivesse tratando apenas dos seres humanos. Lembrando que agora a gente está em um nível muito diferente comparado, né, a tempos remotos, onde os animais eram apenas animais de segurança, de caça. Hoje em dia os animais são membros de família, são tratados como filhos, netos, sobrinhos, têm nome de gente, né? São interpretados, ou seja, ele não está bem, você vê que o latido não está bom, né? Então existe uma indústria, inclusive, envolvendo isso, que é a indústria pet. Hoje em dia você tem creche para cachorros, gatos, peixes, hotéis, você tem toda uma parte de alimentação e por aí vai, né? Então, quando a gente, de fato, estuda os medicamentos, no caso aqui os fármacos, que ainda não chegaram a ser os medicamentos, a gente tem que lembrar que esses animais de laboratório, eles estão fazendo não só pelos humanos, mas também pelos próprios animais, porque esses fármacos que são descobertos, eles também serão utilizados para tratar esses animais em todas as escalas, porque também aquele animal que se come, como, né, vacas, ovelhas e por aí vai, também ficam doentes e também precisam ser tratados. Então, existe uma escala de efeitos sobre essa questão de um bom protocolo, né, do uso de animal de uma forma ética e racional, que perpassa muito além do uso humano, perpassa também a própria vida desse animal. E é por isso que a ideia é que, com relação à rede de biotérios da UFF, dessas atividades educacionais, né, educativas, didáticas, seja lá o nome que a gente der, tem para mostrar que essa área de animais de laboratório, ela tem que ser investida exatamente, inclusive, para chegar na total substituição dos animais. Lembrando que os animais, eles são extremamente importantes, porque, apesar de existirem estudo em célula, os efeitos fisiológicos ainda dependem de todo um sistema coexistente, que é o ser vivo, né? No funcionamento total de todos os seus sistemas cardiovascular e por aí vai. Então, enquanto não tivermos uma substituição desses sistemas fisiológicos, infelizmente, todos os animais, inclusive os homens, né? Porque aí vem homens também, os seres humanos também são animais, entre aspas, de laboratório, mulheres grávidas, crianças, idosos. Então, todos nós, no fundo, não deixamos de ser animais de laboratório quando participamos de pesquisa. E, por isso, a importância do protocolo. Então, agradecer novamente à professora Célia e todos da mesa que participaram da abertura. A professora, a doutora Estela já está aí, mas antes dela entrar, porque eu vou colocar ela, né, eu vou chamá-la exatamente às 10 horas para cumprir, ela é do Reino Unido, né, britânica, então existe um horário já pensado, ela vai começar às 10, como eu prometi. Eu não sei se vocês tiveram tempo de olhar o site do NC3R. Eu vou passar aqui só para vocês poderem ver. Eu não sei se todo mundo teve chance de visitar o site, então eu vou abrir aqui para vocês esses poucos minutos, que eu acho importante divulgar. Eu vou colocar aqui no chat, caso vocês queiram entrar também para ver. Vou colocar no YouTube, a gente está ao vivo no YouTube também, no canal da Coordenação de Pesquisa, que é a coordenação que eu coordeno aqui na Universidade Federal Fluminense. Eu vou parar meu compartilhamento e começar o compartilhamento do site. Só um minuto. Vou ver se ele vai compartilhar. Não sei se vocês estão conseguindo ver.
Eu vejo.
Sim.
Então, mostrando aqui o site, só para aqueles que não conhecem ou não tiveram tempo de entrar no site. Então, o que é o NC3R? É um centro nacional que envolve a questão da pesquisa utilizando os animais de laboratório e com foco nos 3 Rs, né? Então, esses 3 Rs, eles visam exatamente esse uso ético, a diminuição, a substituição, a redução e toda essa compreensão de que o futuro ainda será, né, nós esperamos que em breve seja a substituição total por métodos alternativos, assim que a gente puder fazer isso. E é por isso que a questão da formação é um foco da UFF. Precisamos de mais pessoas engajadas nessa área para que a gente possa, de fato, atingir isso que a gente quer, que é substituir totalmente animais, incluindo o homem. Essa questão da substituição ela é uma questão que não é simples, a gente já falou sobre isso. Tem uma questão de preço também, lembrando que a pesquisa no Brasil ela é totalmente, né, ou quase que totalmente subsidiada pelo governo, então existe um custo aí. Esse custo em relação aos reagentes também, que são obtidos fora. Então, quanto mais pessoas nós tivermos envolvidas para a melhoria desse sistema, inclusive com relação à diminuição do preço e do custo desses ensaios, melhor para toda a sociedade, e aí, quando eu digo a sociedade de todo o mundo, não só brasileira. Dentro desse site, vocês vão ver vários itens de interesse para a gente. Eu não sei se a doutora Esther vai tratar mais disso, eu só estou aqui falando em português, ela vai falar em inglês, então, uma das coisas que eu quero lembrar vocês é que o Google ele tem legenda, então vocês podem ativar a legenda, como ela é britânica, a probabilidade que a legenda fique muito boa, porque acaba que essa legenda é feita para inglês, então, provavelmente, essa legenda vai ser o suficiente para ajudar vocês. De qualquer maneira, está sendo gravado esse evento e a ideia é colocar, inclusive, legendas para poder disponibilizá-los, tá? E isso vai ser informado a todos os inscritos e os interessados por e-mail, tá certo? E a gente vai divulgar também na nossa página da coordenação de pesquisa, nosso portal de pesquisa da UFF. Então, vocês vão ver que tem várias questões, inclusive questões relacionadas a financiamento, a relação da divulgação para a comunidade, né, para todos, e outros detalhes que eu deixo aqui, só essa visão geral para vocês, porque eu acho que é interessante que vocês, aqueles que estão aqui, tenham interesse, eu sei, por essa área, né, estou aqui, escolheram participar desse evento, e aí depois gastar um certo tempo sobre isso, e lembrar, agora vocês têm o nosso contato, e lembrar de que nós temos interesse em trabalhar em conjunto, em formar uma rede com outras instituições e formar mais pessoas, ou seja, se tiver alunos interessados em pós-graduação na área, também podem nos procurar, porque a UFF tem interesse em formar o maior número de pessoas possíveis que sejam multiplicadoras dessa visão dos métodos alternativos, da substituição e da redução. Tá? Eu vou parar aqui de apresentar. Eu vou ver se a professora Esther se a doutora Esther já está pronta para começar. Hi, Esther. Can you begin already? Oh,
yes.
Hi, I can see you.
Yes, I can. Hi.
So,
I'll just go ahead and share my screen.
Oh, thank you a lot. Thank you for this opportunity to have you here. Almost here. But this is the best thing about the pandemic stuff is that we can have you without, bring you here so fast, okay? Thank you a lot anyway. The idea, I told the people in Portuguese that the idea for us is to have these meetings on the next year, yes, on 24, with lectures that bring not only quality, but more information, yeah, about the use of animals and everything that can help us with this approach that your mission, yeah, in your center, okay? So I give you the space for you. It's all of you. It's all for you. So you do whatever you want to. Okay, you are very welcome. Thank you.
Excellent. Thank you.
Thank you.
Yeah, so I'd like to thank you very much for inviting me to talk to you today. And my name is Esther Pearl, and I am going to explain how we can design rigorous experiments and how the Experimental Design Assistant, our online tool, can help. But I thought to start with, I would just give you a little bit of background, just why I'm talking to you. So I come from the UK's National Centre for the 3Rs, and we are an independent scientific organisation, but we are primarily funded by the UK government. And our goal is to advance the 3Rs, so reducing animal use, and when animals have to be used, improving their welfare. We work internationally and nationally with research community, including funders, journals, regulators, and scientists in both academia and industry. And most of our budget goes to funding 3Rs research, so to develop tools and technologies that reduce our reliance on animals. We also have a number of in-house programs led by the scientists in our office, and that includes the one that I am part of, which is focused on experimental design and reporting. So that's why I'm here talking to you. And my talk is going to be split up into 3 parts. So first, we'll talk about the evidence for why we do need to improve our experimental design, and then some key ways that we can improve things, and then finally the resources, including the online tool, that can help us implement those things, so to make improvements easier. So let's start with why we need to improve experimental design in the first place. So you've probably heard of issues with the reproducibility of animal research, and this is a topic which has been discussed at length, and many scientists and organizations are concerned about this, as you can see from these headlines over the last few years. The reason we're all concerned is that if published experiments are not reproducible, we can't rely on their results, and it's not clear how they can contribute to scientific progress. So animals are being wasted in experiments that are ultimately not informative, and we are working to reduce this. So to find out the scale of the reproducibility problem in preclinical research, a group started the Reproducibility Project Cancer Biology. So this project began in 2013, and it was inspired following the publication of reports from the pharmaceutical industry that suggested between 80 and 90% of published research couldn't be reproduced. So this project, the Reproducibility Project, wanted to replicate 193 experiments from 53 high-impact cancer biology papers. So the papers they were looking to replicate were published between 2010 and 2012. So the project finished in 2021, and overall the results were not that reassuring, unfortunately. First, the scope of the project had to be reduced. So out of the 193 experiments, they found that they couldn't complete 143 of them. So they could only complete about 26% of what they set out to complete. And the reasons for this were mainly because there was methodological detail missing in the original papers, and that materials such as mouse lines were not available. Then they found out of the 50 experiments that they could conduct, about 41% of the findings couldn't be replicated. So this is where the effect in the original experiment fell within the 95% confidence interval of the effect in the replication. So that only happened in less than half. So the graph at the bottom of the slide shows the density plot of the effect sizes in the original, so that's the top one here, and then the replication experiments. And we can see that in the original experiments, there were some large effects of treatments or interventions they tested, but in the replications, the effects on the whole were much smaller, including some negative effects for some of the treatments that they used in experiments. So all of these studies that are not reproducible contribute to research waste. And The Lancet published a paper about 15 years ago estimating that 85% of our investment in biomedical research is wasted. And more recently, a study came out looking at ecology research and had similar conclusions. So this more recent study reviewed 33 meta-studies in ecology, and that covered over 10,000 studies in total. And they found that between 82 and 89% of research was wasted. So 82% was their best case estimate. So this was mostly down to issues with poor experimental design, incomplete reporting, or in some cases, studies that were just not published at all. So if we flip that number on its head, that means that only 10 to 15% of research that was funded was actually usable, and that's a really low return on our investment. But when the research involves animals, we have additional ethical implications. So in the UK, like most of the world, the authorization for animal experiments is given based on a harm-benefit analysis. So the harms to the animals that are inherent in their transport, their housing, or a consequence of the scientific procedures they receive are weighed against the likely benefits to science and society, such as new scientific knowledge or improvement in human or veterinary health. But if an experiment is not designed carefully, for example, if it's underpowered or a risk of bias hasn't been addressed, then the results won't be reliable. Or if it's not reported properly, then no one can actually use the results, so the benefits can't be realized. And this means that the research is unethical. So studies like this are a waste of animals, but there are also implications when entire programs of clinical work are based on findings of unreliable animal research. So irreproducible findings in the scientific literature can trigger clinical studies and subject patients to treatments unlikely to be effective, but they can also delay the discovery of effective treatments. So improving the rigor and transparency of animal research will help us improve the entire drug development process. And irreproducible research is also an ethical problem, but also a financial one. So this paper was published in 2015 and looked at how much irreproducible research was costing in the US. The budget for preclinical research at the time was $56 billion a year, and with a very conservative estimate, they thought about half of that was not reproducible. So that meant that $28 billion a year was being wasted in the US, and that's a huge amount of money which could be put to better use. So many of the root causes of reliability and reproducibility issues have been identified. So this report, this image is from a report from the Academy of Medical Sciences, and this report was done in collaboration with the major UK biomedical research funders, and it highlights the 6 main causes that most groups have found when they're looking at this. So I'll just quickly go over these. So one issue is omitting null results. This is also known as publication bias. So this is when scientists or journals decide not to publish studies unless the results are statistically significant, and this ends up biasing our literature. There's also a problem with data dredging, which is also known as p-hacking. So this involves repeatedly searching a data set or trying alternative analysis methods until that magical statistically significant result is found. Then there are problems with weak experimental design. So if a study has methodological flaws, it can mean it's unlikely to ever produce reliable or valid results. There are also issues with underpowered studies. So power is the probability that you will detect an effect if it actually exists. But when a study is underpowered, then it is too small to reliably indicate whether or not the effect exists. So you may as well not have done the study. Then there are issues with errors, so human errors, such as you misidentify a reagent or you make a computational error. And then the last cause they found was underspecified methods. So even when a study is really robust, if the methods aren't shared with other scientists in enough detail, then the methods can't be replicated, the results can't be verified. And we saw this in the reproducibility project cancer biology slides I presented. Okay, so we know that there's evidence there's a problem. What are some ways that we can improve rigor? What are some key things that we can do? So there are a number of measures we can use to make sure our experiments are well designed and rigorous. One such measure is randomization, and this is used to help us reduce subjective bias. So when we allocate animals to treatment groups, the method of randomization is really important. So haphazard is not random, so that's where we as humans think we're being random. We actually need to use a validated procedure to randomize animals into groups. So a computer-generated list of random numbers, throwing a dice, flipping a coin, essentially something that we as humans can't subconsciously influence. So randomization has 2 main purposes. So it helps minimize selection bias and reduce systematic differences in the characteristics of animals allocated to treatment groups. For example, haphazard selection may mean we get all the slowest mice in the same group because that's just how they were caught. Randomization also helps meet one of the key assumptions of statistical analysis, which is that different groups should be drawn from the same background population using random sampling. So technically, if we have not randomized animals to groups, we shouldn't be running a statistical analysis on the data. So the graph on this slide really demonstrates the difference between haphazard and random, and it's based on a simple internet poll. Human participants were asked to pick a number between 1 and 20, and the results were compared to a truly random allocation using a computer. There were 20 numbers, so if it's random, each number has a 5% chance of being picked, and that's this dashed line here across the graph. And this is what we can see with the computer results. So the computer results are the white bars, and they oscillate around the 5% line. But when human participants picked a number at random, about 17% chose the number 17, and about 12% chose the number 7. So there might be a cultural explanation for why these particular numbers were picked, but the point is that humans are not very good at being random. We're a bit rubbish at it. So randomization needs to be done with a validated method that we can't influence. So we can see the impact of randomizing or not randomizing in the published literature. So these findings are from a systematic review looking at the efficacy of interventions in vivo models of multiple sclerosis. So researchers combined the results from 126 different studies, and they were testing the efficacy of 36 different multiple sclerosis drugs or potential drugs. And they compared in this study studies that used random allocation to groups versus studies that didn't. And we can see we've got on the y-axis here the effect size, so the percent improvement in neurobehavioral score with the drugs. And in the studies where random allocations to group were not used, the effect size was on average about 42%. But when random allocation to groups was used, the effect size found by those studies was only about just over 20%. So this is showing in this case, non-randomized studies reported a treatment efficacy twice as big as what it really is. So studies that don't randomize overestimate treatment efficacy. And this also shows that this is a problem with the design of the research, not just the reporting, because if randomization had been used properly in all studies, just not reported in some of them, we wouldn't see that difference between the effect sizes on average between those two groups. So we need to be careful when we're allocating to groups that we use randomization, but we also need to think about how to avoid introducing bias throughout the experiment. So once our experiment is up and running. And some of these things that can bias our experiments, we can get help from animal technicians and animal husbandry staff. So do talk to your animal facilities and ask for their help, because they can really help you reduce bias in your studies as well. So some things to consider are when you administer treatments. For example, don't administer all of your treatments in the morning and all of your controls in the afternoon, because you could be looking at the effects of the time of day as well. When you are taking measurements, make sure that you measure all of your animals on either the same piece of equipment, or if you have to use two different pieces of equipment that are essentially the same model, make sure you've got some treated and some control measured on each piece of equipment, because calibration differences between the two could introduce bias if you measured all your controls on one piece of equipment and all your treated on another. Also, have a look at how you're taking measurements or performing health checks. Are you doing them in the same order every day? Are you randomizing that order? And then think about where your animals are placed in your facility. So in this graphic on the right here, we have all the treated animals in the top row of the rack, and they are exposed to more light than the control animals, which are one row down. So that's introducing a bias there as well. And also think about the levels of training of staff handling the animals. So for example, if you're training new staff and all animals in the treatment group are injected by someone who's already trained and competent, and all control animals are injected by the trainee, that could introduce bias in the way the animals are handled and the way the injections are performed. So we've got to be careful about introducing bias once the experiment starts as well. Another measure we can use to help reduce bias is blinding, also known as masking. And this is where researchers don't know what experimental intervention the animals have received, and it allows us to remain objective. So this study here is over 50 years old, but it's a really good demonstration of why blinding is important. So the experiment was carried out on two populations of rats. One population was described as bright rats and the other as dull rats. And the rats were put through a memory test. So it was a T-maze, and the T-maze had a white arm and a dark arm. And in the training phase, when the rats went to the dark arm, they got a food reward. So they got rewarded for making the correct turn. Then in the experiment, this was carried out over several days, and researchers just counted the number of turns to the correct arm. You can see in the graph that the bright rats started off better than the dull rats, and they improved more over the course of the study. And by the end of the study, the difference between bright and dull rats was statistically significant. The point is, though, that the experiment was not actually performed on the rats, it was performed on the researchers. There was actually no difference between the two populations of rats. They were labelled bright or dull randomly on the morning of each day of the experiment. So the only difference between those two populations were in the expectations of the researchers. And the researchers said that they thought the rats in the bright group were brighter, cleaner, tamer, and more pleasant than the rats in the dull group. And researchers working with the bright rats said that they were more relaxed and casual around the bright rats. So these differences we see on the graph are due to differences in the researchers' behavior towards the rats, rather than something inherent to the rats themselves. And this is why blinding is really important, because if we're not blinded, we can unconsciously influence the behavior of our animals and then influence our results. So this experiment is another one that shows the importance of masking or blinding. So this involved 157 veterinary medicine students, and they looked at 5-minute video clips of pigs in their pens and scored how frequently the pigs showed positive or negative social interactions. And they had two separate groups of pigs. They had control pigs and pigs called high social breeding value pigs. So they were basically better behaved pigs. And you can see in the graphs at the bottom, the bars on the right represent the high social breeding value pigs, and they had more positive social behaviours than the control pigs and fewer negative social behaviours than the control pigs. But what is actually interesting is it turns out the footage of the pigs was actually the same footage that had been visually manipulated to make the students think they were observing two different groups of pigs. So just like in the cartoon at the top here, the footage for the high social breeding value pigs was mirrored, the brightness changed, and dates and pen numbers were kind of added to the videos to give the misleading perception that the students were looking at two different videos of two different groups of pigs, but it was actually the same video. So half of the group was told the first video they saw was the better behaved pigs, while the other half was told it was the second video. And so the differences the students observed were entirely the result of their own expectations. So this highlights the importance of masking or blinding when we're measuring the outcome of an experiment. So this helps remove our expectations and subjective bias. So I just wanted to go through some ways that we can implement masking, some of the more easy ones. So you could ask someone to code your animals or samples for you. So for example, if that pig study was a genuine study into pig behavior, you could have someone code the videos. So you were just counting the behaviors without having any expectations about what you were going to see in each video. You could also ask somebody else to give the intervention or take the measurement for you. So for example, if you are doing an IP injection of either a drug or a control, you could load all the syringes and label them for which animal each syringe goes to based on your randomization sequence. But you could give somebody else the syringes and they know which animal they need to inject with each syringe, but they don't know the contents of the syringe. So they're going to be just as careful with all of the animals and all of the syringes. So that takes out the subjective bias at that stage. When you're analyzing data, you could ask someone to code your groups of data so you know which values go together, but not which group had which intervention. Or you can use an independent analyst. So you ask somebody else to conduct the analysis for you. So I'm going to give a PDF version of these slides to Helena to share with everybody after, and because there are a lot of hyperlinks on the slides, including these ones here. So we have a link here to the NC3Rs resource page on masking. So we interviewed some researchers to find out what kind of masking they were implementing in their studies and what kind of barriers they found. And we've got a research page with some strategies for masking different stages of experiments. And the British Pharmacological Society has an animated video to introduce the concept of masking and an e-learning module about masking. So for the e-learning module, it is free to use, but you just have to register for a free account on their website. So to round off our talking about randomization and masking, I want to talk about the impact of both of them in the literature. So this study was an analysis of 290 abstracts describing animal research presented at the meetings of the Society for Academic Emergency Medicine. And they looked at whether the use of randomization and blinding was mentioned in the abstract and whether the studies reported positive or statistically significant results. So this graph here shows that when abstracts reported both randomization and blinding, about a third of them, just under a third of them, found statistically non-significant or negative results. When abstracts only mentioned one, randomization or blinding, about 1 in 5 reported statistically non-significant results. But when abstracts mentioned neither, it was only about 8%. So less than 1 in 10 reported statistically significant results. So this difference is statistically significant, and studies reporting neither randomization nor blinding are 5 times more likely to report statistically significant results than studies that reported both. So this means that the results of studies that don't report randomization and blinding are much more likely to be false positives. And it also shows this is unlikely to be a reporting issue because if randomization and blinding had been used in all of them, just not reported, we wouldn't expect to see this difference between the different groups. So on to another bias that can affect our results, and that's sex bias. And so I just wanted to spend a couple of slides to show why only using one sex is a problem. So at the moment, most preclinical research is conducted in males rather than females, or rather than both sexes. And this is a problem because we can't extrapolate findings from males to females. This used to be an issue for clinical trials, but about 30 years ago, there was a real push to include women in trials as well. And nowadays, a typical clinical trial, you'd expect to have half and half, male and female. But this hasn't happened for cell and animal research yet, and sex is often not considered as an important factor in the design, analysis, or reporting of preclinical research. And this is a real missed opportunity for us to collect crucial information And this is information that could inform the design of clinical trials or provide warnings on adverse effects that are sex-specific. So an example of this, and why assuming male and female responses are the same is dangerous, is the insomnia treatment drug Zolpidem. So 20 years after its release to market, the US Food and Drug Administration issued an alert that to reduce the dosing for women by half because of risk to women for next morning impairment. So they were still feeling the effects of the drug the next morning. And this adjustment to dosing arose because of sex differences in clearance rates. And these are things that would have been detected if pharmacokinetic studies had been carried out in both sexes prior to clinical trials. And this isn't an isolated finding, unfortunately. 8 out of 10 drugs withdrawn from the clinical market in the US between 1997 and 2000 were withdrawn due to significant health risks for women. So most biomedical research funders do require a full justification for the sex of animals to be used in grant applications. And a good example is the NIH in the US, and they state that using both sexes should be the default for preclinical experiments, and that sex should be taken into account in the design, the analysis, and the reporting of research. And funders in the UK are bringing that in as well. So I just wanted to highlight the problem of sex bias in animal experiments by showing you this graph from the journal Pain. So this is an analysis of all primary research articles published in the journal Pain. And if you look in the areas of blue, these are studies that used males only. And we can see that it used to be about 80% of studies using males only, but by 2019, that was down to 50%. But we still have 30% of studies only that are using both sexes. And the kind of worrying thing is this is in the field of pain research. So this is an area where we know there are differences between male and females in terms of pain pathways. And clinically, we know most patients with chronic pain are women. So this is a field where we know there are sex differences. But let's focus in just on the studies that did use both sexes. So that's areas in orange and yellow and actually investigated the effect of sex. So that's the orange and yellow fields. And we can see that about half of these studies actually did identify differences between the responses of male and female animals. So that's the yellow shading. So this is information that we will completely miss if we used only one sex or if we included both sexes but didn't actually look for differences. So we are missing important biological information if we only use one sex. We have the same sex bias in cell-based research. So this paper looked at in vivo and in vitro research, but for today, we're just going to focus on the in vitro side of the slide. So most papers didn't report the sex of the cells used for in vitro research, and of those that did, over 70% were using male cells. So this bias towards male animals and cells, they found it occurred even when the disease being studied was actually more prevalent in women. So what are some reasons why people have been preferring to use male animals? So historically, one of the reasons was that they believed variation in female animals would introduce extra variability into their data. For example, variation caused by the estrus cycle. But we do have empirical evidence that this is not the case for many of the outcomes we measure. So this graph here shows variability data from nearly 300 studies, and they were looking at behavioral, morphological, physiological, and molecular traits in both male and female mice. So the female data was collected without considering the estrus cycle stage. And on the graph, female data was plotted in red, male in blue, and where it overlaps, it's purple. And you can see that most of the graph is purple. And this is because the male and female variability pretty much completely overlapped. And for some traits, the variability was actually greater in males than females. So differences in variability is not a good reason to exclude females from animal experiments. And if we use both sexes, we have the possibility of our findings being generalized to the whole population, whereas if we use only one sex, we can't. And also, if there is a big difference between the sexes, if we are using both sexes, we can detect that, whereas if we only use one, we'd never know that's the case. Another common misconception was that using both sexes in an experiment requires double the animal numbers, but this isn't always the case. So using both sexes does have implications for the design and analysis of the experiment, and the sample size you need actually depends on the purpose of your experiment. So what conclusions do you want to draw? So the first thing to to remember is that males and females should be randomized into different experimental groups using separate randomization sequences. So that means you'll end up with the same proportion of males and females in each group, because if you don't, then you've got a sex bias in your groups. You also want to run your experiments together, so males and females as part of the same experiment. You don't want to run your experiment in one sex and then wait a couple of months and then run it in another sex, because if you do that, you can't analyze the data together, and it means your experiment's not as sensitive. So then, if the purpose of your experiment is just to determine the overall effect of your treatment, and you're not specifically investigating sex differences, you just want to make sure that your results apply to both males and females, then that's the box here on the left. You don't need to increase animal numbers. You still need a power calculation to work out the number of animals per group, but just make half of the group male and half female. And then use sex as a blocking factor in your analysis, and that allows any variability explained by sex differences to be taken into account, and it increases the chances of you detecting the effect of the experimental intervention you're interested in. On the other hand, if you are interested in investigating whether the effects of your experimental intervention do depend on sex, then that's the right-hand side here. You need to use sex as a factor of interest in your analysis, and you do need to power your experiment accordingly. And if you are doing post-hoc group comparisons, this might mean using more animals compared to a single sex study. So this is a very brief overview, but the link at the top of the page links to more information on the EDA website about guidance for including both sexes. So now we've kind of covered why we need to improve experimental design, some of the things we should be considering to improve the rigor of our experiments. Now I want to talk about the resources that we can use to help us. So what are some things that can make this a bit easier for us? So first, I'd like to signpost you to the experimental design resources you can find on the NC3Rs website. So if you go to nc3rs.org.uk/3rs-resources, you come to our resource library. And then we've got a little bit here for experimental design and reporting resources, and these are just a few of them here. So we have links to our blinding or masking resource that I mentioned earlier. We have a webinar on best practice in experimental design. We have guidance on conducting a pilot study, a webinar on using both sexes in animal experiments. But today I'm mainly going to focus on the Experimental Design Assistant, our online tool to help you design experiments. But I will touch on other useful resources at the end. So the Experimental Design or Assistant, or EDA, is an online tool. It's got a supporting website, and the aim is to help us as researchers design our in vivo experiments well. The EDA was developed by experts in experimental design and statistics, so we've got a big working group behind it, and it is currently used by over 16,000 researchers worldwide, and it's available at eda.nc3rs.org.uk. So the EDA website has trusted guidance and information about experimental design, and both the website and the tool focus on internal validity, so how to make your experiments more likely to give reliable results, and the entire resource is free to use. So using the EDA is a bit like having a statistical assistant help you design your experiment. So it provides tailored advice on how to improve the design of the experiment. It can suggest statistical tests that are compatible with your specific design. We have guidance and power calculators to help you determine the appropriate sample size in your study. We have dedicated support for randomization and blinding or masking. And the EDA enables you to share your experimental plans with collaborators, colleagues, funders, ethical review bodies, and in your papers. And I'd just like to also state at this point, the NC3Rs is also a funding body. So we are used to dealing with proprietary information, and we apply the same levels of security to the EDA as we have for our grant system. So your experimental information is safe in the EDA. So inside the EDA, each experimental plan is represented as a diagram made up of these colored shapes called nodes, and each node represents a part of your experiment. And we have 3 different types of information in an EDA diagram. So there's high-level information about your experiment and its objectives. So this is things like your null and alternative hypothesis, what's the main effect of interest. We've got information about the practical steps. So these are the things you'll physically be doing in the lab. And then information about the analysis plan. So I'll just quickly talk you through this experimental diagram just so you can find your way around it. So in this example, we start with a pool of animals. They're allocated into 2 groups, in this case by complete randomization. Animals in group 1 receive injection of vehicle, in group 2 an injection of a drug, and then we measure the plasma glucose in all the animals. And then the data collected is used in the analysis, along with information such as how many variables are there in the experiment. For example, in this case, the independent variable of interest, whether the animals received vehicle or drug. So an EDA diagram is more explicit than a text description of an experiment, as there is more information inside each of the shapes. So if we think of blinding or masking as an example, in a text description of blinding, a paper may say the experiment was performed blinded, but that doesn't tell us exactly which steps were masked or blinded and how that was actually implemented. And in the EDA diagram, there are several places you can describe it. You can mention how you're blinding when you allocate animals to groups and start giving them the interventions. You can mention it in the measurement node and also when analyzing your results. And in each of those nodes, there's a drop-down menu that gives you options for different ways that you can blind at those stages. And that helps you decide what's appropriate, but also what's feasible in each stage of your experiment and with the facilities and setup that you have. You can represent any experiment as an EDA diagram, and diagrams are machine readable. So this means that the EDA uses computer-based logical reasoning to provide you with the appropriate feedback. So an example of feedback is this message here. And this is when you design an experiment using only one sex, the EDA gives you feedback asking if using only one sex is appropriate for your experiment and explaining how this will limit the generalizability of your results. So if you were planning a single-sex study and then decide to alter your experimental plans and include both sexes, then the EDA provides guidance to help make sure you get the most out of the information that you're generating from the experiment. So this includes helping ensure you have the same proportion of each sex in each group. It helps you decide how to take account of sex in your analysis and what implications that decision has for your sample size calculations. So the system includes other features to help researchers use rigorous methods, and this includes generating randomization sequence to allocate animals into groups. So the EDA can do complete randomization, block randomization, and randomization within factors. To enable blinding or masking, the EDA emails your randomization sequence directly to the person helping you with masking, so you don't see it and you can remain unaware of the group that each animal has been allocated to. And the EDA can also help with sample size calculation, and I'll come back to that in a moment. So let's focus on how the EDA helps with randomization and masking for now. So how can the EDA help? So the randomization sequence generated by the EDA is in the form of an Excel spreadsheet, and that gets emailed to the person who will help you with randomization and blinding. So this is the person who will be aware of the allocation of the experiment. So this is an example spreadsheet for an experiment where animals are allocated into two groups. So we can see here in column C, there's group 1 and group 2 mentioned. We can also see that there are different tabs at the bottom for male and female. And this shows that the EDA has generated a randomization sequence for each sex separately, and that makes sure we have the same proportion of each sex in each group. So column C is what has been randomized. So the order that the groups appear in, group 1 and group 2 appear in here, has been randomized. Column B is where the unique identifier information for each animal is put by the person who is not blinded. So for example, these cartoon mice on the side here, they represent 8 males that need to be allocated into the groups. So the person helping you with the allocation, they'll add the animal ID. So in this case, let's say it was a numerical tail tattoo. They'll pick up their first animal, add the ID into column B, and know that animal's allocated to group 1, the second animal to group 2, the third to group 1, and the fourth to group 1, etc., until all 8 males have been allocated to the groups. They will then do the same thing with the females by clicking on the female tab. And then they save the spreadsheet and they don't share it with you until the experiment is finished. And that means you can remain unaware of group allocation and reduce bias in your experiments. So if you haven't included enough information regarding what you're randomizing and how, the EDA will provide you with guidance to help you decide what to do. And the same goes for blinding. If you haven't provided enough information about blinding or masking, the EDA will provide you with support for that. And then there is also guidance on the EDA website. So the previous slide was feedback you get within the tool, and then this is an image from the EDA website. So you can get help with experimental design without logging into the system as well. So now let's talk about how the EDA can help you determine an appropriate sample size. So the EDA itself has power calculators for both paired and unpaired t-tests, and we also have a lot of advice on the EDA website, including why power calculations are important. We have a decision tree to help you decide the most appropriate power calculator to use for your particular experiment. The power calculator itself is also available on the EDA website, so you don't have to log in to use it. And the website has information to help you identify the parameters needed for your calculation. So help identifying a biologically relevant effect size, or help estimating the variability. And this helps you get the best estimate of the number of animals or experimental units you need in your experiment. So once you have designed your experiment in the EDA, you can share your plans with others, and there are a few ways to do this. So one way is to generate a PDF report, and this is a date-stamped document, and it contains the key experimental design information in a standardized format, and then it has an image of your EDA diagram at the end. So this key information is listed here. This is the information that your funder or ethical review board want to know about your experiment. And the PDF report can be used as kind of an aide-memoire when you're preparing a grant or ethical review application. So all of the information you need is in one place, and you just need to make sure that it's made it into your application somewhere. You can also use the PDF report as supplementary information with your manuscript to provide extra detail on your experimental plans. The PDF report does have any feedback from the EDA that you've not addressed. So if you do create a report, you might get reminded, for example, that there's some information missing. Then you can go ahead and fix that and then generate a new report. You can also share your experiments using a read-only diagram. So this is a static online version of your EDA diagram, and it can be shared with anyone, whether or not they have an EDA account. So it has the same table of key experimental design information on the left here as the PDF report, but instead of a static image of your EDA diagram at the end, there's a clickable version. So by clicking on this little double arrow here, you can hide the table, and then the person you share this with can click on all the different parts of your experiment to get more information about the part they're interested in. For example, if they want more information about your pharmacological intervention, they'll click on this node here and they'll get extra information. So the read-only diagram is shared by an unguessable URL, and there is an access code on that. So you, as the owner of the diagram, can remove the access code when you want your experimental plans to be more publicly available. For example, if you've included the link as part of the methods in a scientific paper, you can leave the access code on while the paper is being peer reviewed and remove the access code when it's published, so that anyone reading your paper online can click on the link and get more information about your experimental design. If you do want to see the EDA in action, we run regular live demonstrations via Zoom. So this link up the top, nc3rs.org.uk/EDA demos, is where you go to register. The next live demonstration is in January next year, but we also have one scheduled for March. But we also have recorded demonstrations on the EDA website. So that's the bottom link here. And both the live and recorded demonstrations cover how to build your diagram in the EDA, so how to use it technically, how to get feedback and use that to improve the design of your experiment, information on how to generate the randomization sequence that can take into account the factors that you need to randomize separately, how to create both the PDF and read-only diagrams to help you share your plans with others, like funders, ethical review bodies, or to use in manuscripts, and how to to find the help that we've built into the system so that you can figure out if you do get stuck. So I just wanted to touch on a couple of other useful resources before I wrap up. So as I mentioned earlier, funders are increasingly expecting that preclinical research uses both sexes and that any single-sex studies must be really carefully justified. So this will be new to quite a few people. So I just thought I'd list some resources that can help. So obviously we've seen that the EDA can help by providing you guidance if you use that to design your experiments, but we also have guidance just on the website. We also hosted a webinar with the UK's Medical Research Council last year on using both sexes in animal research. The recording is available on our website, and it includes addressing barriers to including both sexes and husbandry and phenotyping considerations, etc. And the NIH in the US has created some resources to help us integrate sex into research as well. Another resource I'd like to mention is the ARRIVE guidelines. So this is another really important tool to help us improve reproducibility of animal research. So the ARRIVE guidelines are a checklist of key information to include in a scientific paper, and that helps us describe the study in a comprehensive manner so our research can be evaluated adequately and the methods and findings can be reproduced. We released an updated version in 2020. And the ARRIVE guidelines do apply to all in vivo research. It doesn't matter if the species used doesn't require ethical approval, because the ARRIVE guidelines are about rigorous science. So that does apply to any species. Now, obviously, if we're coming up with international standards and guidelines, we need to make sure that it works for the entire scientific community. So the ARRIVE guidelines were developed with input from the community really carefully built into the process. So we had an international working group, and we had expertise from across the life sciences, including funders, journal editors, statisticians, methodologists, and researchers in both academia and industry. We also used a Delphi exercise with external stakeholders to maximize the diversity in fields of expertise and geographical location. So we ended up having expert input from 19 different countries. And we conducted formal road testing with researchers who were preparing manuscripts to ensure that our guidance was useful and well understood. And we also considered other reporting standards to ensure that we didn't provide conflicting advice which would be confusing to researchers. So ARRIVE 2.0 are compatible with other guidelines that have been published in recent years. And because they provide a comprehensive set of recommendations specifically tailored to description of animal research, if you use the ARRIVE guidelines to write a manuscript with animal research in it, it makes sure that you're complying with the more general guidelines and journal policies as well. So the ARRIVE guidelines contain 21 items split into 2 different sets. So if you're not used to reporting all of this information, then focus on the essential 10 initially. So the essential 10 is the basic minimum to include in a manuscript, because without this information, readers and reviewers can't assess the reliability of your findings. And then the recommended set provide really important context, and they really complement the essential 10. So reporting all of them is best practice, but if you do have to focus on just some, then do focus on the ARRIVE Essential 10. The guidelines come with an explanation and elaboration document, which was published alongside the guidelines in PLOS Biology, and it's also available on the ARRIVE website. So this explains the rationale behind each item in the guidelines, why it's important to report the information, and it also provides examples from published literature showing how to report this information well. But really importantly, it clarifies key concepts, particularly around experimental design and statistics. So the EDA and ARRIVE are really complementary, and the EDA helps you design experiments and consider upfront all of that information you need to report in your publication to be in compliance with the ARRIVE guidelines. So I do recommend using them together. And in fact, this slide shows how ARRIVE and the EDA and some other resources complement each other throughout the experimental cycle. So obviously, when you're planning your experiment, you can use the EDA to design your experiments, but the ARRIVE guidelines can also help you, especially that explanation and elaboration document, because there's recommendations on experimental design, minimizing bias, sample size calculation, statistical analyses, etc. So if you use the 2 of these together, you can design really robust experiments. Once you've planned your experiments, we really encourage you to pre-register them. There are specific registries for animal research, such as preclinicaltrials.eu and the Animal Study Registry. You can also do this via the journal you're going to publish in, using a format called registered reports, where you pre-register your study and you get feedback on the experimental plans, and then you get published regardless of whether your results are statistically significant or not. Or you can use more general repositories such as the Open Science Framework. And you can use the PDF report generated by the EDA as a sort of soft pre-registration, because it is time-stamped, or date-stamped rather. And you can actually upload the PDF report into the Open Science Framework, or put the link to the read-only diagram there and use that as a form of pre-registration. While you're conducting your experiment, if you have the ARRIVE guidelines next to you, then this makes sure that you are writing down that key information that you need to be recording about your study methods, etc. So you've got that information when you come to write it up. To make sure your data are accessible, you can put your data or figures in Figshare, Zenodo, or the Open Science Framework. And when you're writing your manuscript, obviously the ARRIVE guidelines are really helpful here. to make sure that you put all the relevant information in your manuscript. But the EDA is also useful. If you've designed your experiment with the EDA, most of the information for the Arrive Essential 10 is in your EDA diagram. So it can help you write up your study. Excuse me. And finally, when you're reviewing manuscripts, you can use the Arrive guidelines to help you see if the manuscript you're reviewing has all that key information you need in order to be able to tell if it's reliable or not. And just before I finish up, I just want to mention the River recommendations. So these are 6 recommendations that represent top priorities to improve methodological rigor and reliability in vitro studies. The recommendations are in 3 themes, so experimental design, procedures and materials, and data handling, accessibility and visualization. We have released the River recommendations as a preprint, and in the new year we will start road testing with in vitro researchers. So if you are interested, then you can scan the QR code to find out more and get involved in the user testing study. And I just wanted to wrap up by showing that the Arrive guidelines and EDA are really endorsed by a lot of the major stakeholders in scientific research. So over 1,000 journals recommend the Arrive guidelines in their instructions to authors, and that includes the big ones like Nature and Cell and Science. And many major funding organizations do recommend that researchers use the EDA when preparing a grant application that contains animal research, and they expect that animal research conducted with their funding is reported in compliance with the Arrive guidelines. And this includes the major biomedical research funders in the UK, obviously. We also have grassroots supports from researchers themselves. So the EDA has, as I mentioned, over 16,000 registered users. And over the past few years, we've had over 40,000 copies of printed copies of the Arrive guidelines requested from 50 different countries. And we are actually making translations of the Arrive guidelines. So on the Arrive website, we do actually have the Arrive guidelines in Portuguese if you'd rather read them in Portuguese. So I'd like to thank you very much for your attention and then move on to questions. So we did have a few questions submitted before we started. And so I just thought I would address some of those. So one of the questions submitted was asking about males and females and sexual dimorphism for the data. So if you've got sexual dimorphism in the data, is that against the principle of reduction? So I would say no, because if we think about the contemporary definition of reduction is appropriately designed and analyzed animal experiments that are robust and reproducible and truly add to the knowledge base. So if we're using studies that have both sexes, we get more information from them than we would in a single sex study. So this data is more useful and it's adding more to our knowledge base. And reduction is about designing experiments well to give us knowledge we can use. So if we're only using one sex, then the data is not applicable to another sex and it's not as useful. And also, if we think about it, your sexual dimorphism in the data may be a really important part of the biology that you're looking at. So clinically, we have many diseases that are more prevalent in one sex than another or present slightly differently in males and females. So this could be a really important part of your model. It could be a key biological piece of information. And so I do recommend you have a look at the webinar on using both sexes in animal research, because that covers that a bit as well. One of the other questions was asking about impacts that have already been assessed based on research projects that use methodology that I was going to present today. And I think if we ask about the effects of using the EDA to design experiments, then we haven't actually done a study on this. Unfortunately, we don't have the resources to conduct a full-scale big study while doing the rest of our work. But we do have indirect measures of the effects of using more rigorous study designs on quality of data. So we've seen on the slides I presented on the effects of randomization and blinding and those effects on reliability of data. And if we think about the costs, because that was mentioned in the question, so the costs of poor research are really high. So the slide I presented showing that 50% of preclinical research was irreproducible. So that's half of that money is being wasted. So if we improve rigor in experiments, it can mean that they take slightly more time to do, and sometimes they are a bit more expensive, but not always. And I think the costs of poor research are worse in the long run in terms of our scientific knowledge. We had a question about inclusive science communication to help us develop this subject in the community. So in terms of being kind of more globally inclusive, the resources that we develop at the NC3Rs, they are online for the most part, and especially the EDA is online. It's free to use. I give regular live demonstrations, and I try and shuffle the times I give the demonstrations. So it's hopefully, you know, if the January one is not the right time zone for you, then maybe the March one will work, kind of thing. And we are trying to make our tools available to as many researchers as possible, so it is inclusive, and it's not just for people in the UK. And if we make them free, it's not just for people that have, you know, big research budgets. And scientific research is a real international endeavor, so improving the way we design experiments needs to be as well. So we're trying to come inclusively from that angle, and we're also translating some of our resources when we when we can. So the ARRIVE guidelines, I believe, are in 8 different languages at the moment, including Portuguese. But we also have the mouse grimace scales, rat and rabbit grimace scales. I think they're in Portuguese, and our malocclusion resources in Spanish. So we don't have both languages for both of those, but we are trying to be more inclusive globally in terms of translating some resources when we can as well. We had a question about the impact of using insects in vivo experiments and whether it be possible to replace vertebrate animals or if we'll never be able to replace them. So I think regarding insects, it depends on your specific scientific question. So in some cases, particular questions can be answered in insects rather than mammals. For example, the wax moth Galeria melanella is being used as a microbial infection model by one of the NC3Rs funded researchers. Another example of research we've funded is a lab in the UK using Drosophila to model specific aspects of Alzheimer's disease. So you can use insects for specific questions or to answer specific questions about diseases. But another aspect about replacing vertebrate animals is also using human cells. So if we use organoids or organs on a chip or other forms of 3D cell culturing, using human cells, then this can help us model human diseases, and it might be more translatable than some of the in vivo experiments or some of the animal models. So we've not only got insects to help us reduce reliance on vertebrate models, but we can also use human cells and cell culture to help us reduce reliance on all animals. And there's also a lot of work on in silico modeling, so using computer models to predict results, reducing the number of animals needed. So one of the other NC3Rs funded projects is called the virtual dog. So the aim of this project is to develop a computer model to model different organs and tissues of dogs to predict toxicological endpoints for new chemicals. So at the moment in the regulations, I think you have to use 2 different species to prove how toxic new compounds are. But this virtual dog project is trying to replace the dog part of that so that you can use just rats and the in silico model rather than rats and dogs to test toxicology. And I do think eventually we will be able to replace vertebrate animals in research, but I don't think we're there yet. But I think we are making good progress and we are moving towards that. We also had a question about pharmacokinetics. Can we simulate those without the use of animals? Some people do some pharmacokinetic work in cell cultures first. So they do the first pass in vitro to reduce the number of animals they need or reduce the number of compounds that they take on to further studies. The NC3Rs did also commission some work looking at how accurately human liver microsomes can predict the pharmacokinetics of compounds, and that work has been published already. And as with most experimental design questions, I think the answer is it depends. So it depends on your research question, or in this case, the specific compound. So is the compound something where we have similar compounds that have been tested previously? Or is it a completely new type of compound? Because that might affect how well we could simulate them without using animals. And I think, like the previous question, I think we will be able to simulate or estimate PK without the use of animals in the future, but I'm not sure we're there quite yet, not for all compounds anyway. We also had a question about the impact of detecting murine norovirus in facilities, and does that make animals unable to be used in experiments? And I would say It depends a bit, again, on the welfare implications this has for your animals and on the specific research question. So, for example, if you're using a mouse mutant line that has some immune system issues and your research is looking at, say, a coronavirus viral infection load, for example, then that could interfere with that. But if you're researching something completely unrelated to the immune system, then maybe it's okay. But I would recommend you talk to your vets at your facility with regards to welfare issues, but also perhaps see if your animal supplier has some advice. Or you could check out resources like the Jackson Lab or Charles River, because they have some information about norovirus particularly, I think, as well. And then finally, we had some questions specifically about the EDA. So we had someone ask about different types of experiment and if they could be represented in the EDA. So one was looking at an experiment that has two different groups, and one group has repeated doses, and then animals are euthanized at different time points. And they were asking, can you use the EDA for that design? I'd say definitely yes. And if you have attempted to make an EDA diagram and struggled, then do email us, and I will just put the email address in the chat. So yeah, if you are using the EDA and you do struggle with anything, then do email us. And if you share your diagram with us, we can we can help you sort out and check that you're presenting what you're actually planning to do in your study, and that you're happy, that the diagram is what you're actually doing, and you're happy with the rigor of the experiment. Another question was looking at experiments where you've got two different things you're testing. So in this example, there was a diet, high-fat or low-fat diet, and then animals within each diet were given oral gavage of either a treatment or a placebo. And I would say it's definitely possible to design your experiment in the EDA, but the EDA will tell you at the end to talk to a statistician for your experimental analysis, because in this particular example, I'm assuming the diet is being given to the cage of animals, and then the oral gavage is being given to individuals. So in that case, for the diet, the cage is the experimental unit, because you can't give different animals in the same cage the different diets unless you individually feed them, which most people don't. And the oral gavage is being given to individual animals. So that means you've got different experimental units for different parts of your experiment. So a statistician would help you plan the analysis plan for that. So the EDA at the moment will say you've got more than one experimental unit, so we advise you talk to a statistician. And then the last question was about if you are performing euthanasia on different days. So we talked in the talk about using blocking factors and making sure that you did some of each group on different days and things. And I think the question was, in this case, that increases the data dispersion, so it increases the variability. And they were asking, can you include these blocks in the statistical analysis in the EDA? So if you put blocking factors in your EDA diagram, which I do cover in the demo in the new year, then yes, the EDA will include the blocks in the randomization sequence it gives you, and it will also take account of the fact that you've got blocking factors for the analysis recommendation. It will tell you, for example, you may have to use a one-way ANOVA with blocking factors. And then it will tell you different statistical analysis software that you can use for that analysis, like, for example, InVivoStat, which is free to download, and it is designed specifically for animal research, for the stats to do with animal research. But I would say, with regards to increasing the data dispersion, that's actually probably a good thing for your results, because we have problems in animal research with over-standardization of our studies, and this leads to us getting many statistically significant results that end up being an artifact. So, you know, it's a result that can only be replicated if you do the experiment in a specific institution with a specific researcher. And so this means that the results are unlikely to reflect the actual biology of your question, because humans are really variable. And one suggestion that we've had for why preclinical research doesn't necessarily translate into clinical is that we are standardizing our animals so much and making them so inbred and so exactly the same that we are getting statistically significant results based on an artifact of the standardization rather than the biology of the disease or the treatment we're after. So if we want to solve this, the key is systematically introducing heterogeneity. And if you've got groups of animals and you can't process them all on one day, and you need to do some from each group on one day, some from each group the next day, and some from each group the day after, you are systematically introducing heterogeneity. And by using some from each group each day, you're not adding it, you're not making differences in one particular group versus another. You're spreading that difference across all of your groups. And that means any results that you do actually find are more likely to be the real biology of the question you're asking, rather than just an artifact of the fact that, say, Monday the humidity in the facility was higher, so that's a result of that kind of thing. So by deliberately doing your experiment in batches over time, you're actually introducing some of that natural heterogeneity that we get in human populations. So your results are probably more likely to reflect the underlying biology. So I think that's actually a really good thing, because I think it makes your results a bit more likely to be reliable. So I've noticed we've got a question in the chat about, do we know the impact of the EDA tool for the research submitted to journals that recommend the adoption of these guidelines? Unfortunately, we don't. I would love to know that. I would love to have all of these journals telling me when people submit work with the EDA. I do check. The EDA does have an RRID tag, so I do check on there to see how often the EDA is cited in papers, and I do check when the EDA is mentioned in publications. Unfortunately, lots of people just write in their paper that they've used the EDA, like by typing out experimental design assistant, and they don't cite our papers or the RRID tag. So it can be quite hard to track which publications are using the EDA. But from what I have seen, a lot of people do tend to use it for power calculators, and I've seen a few theses where they've used it to design experiments. But for the most part, people don't cite it well enough for me to track, unfortunately. Most journals recommend people use the ARRIVE guidelines to write up their papers thoroughly, but not all of them recommend using the EDA for designing experiments. But I guess if you're thinking about journals, if somebody has already done their experiment, it's a bit late to tell them how to design it. So, but we are trying to encourage journals to use the read-only diagram. So, encouraging you to put the link to your EDA diagram in your paper to help you design, to help you explain your experimental design more clearly.
Thank you, Esther. Esther, I have a question, not a question. In fact, it's an invitation for you, because I know that we established the time and your time is over. I know. Yeah, you said that 11, it will be the end. I don't know if you have like 10 minutes or something like that, because I know, Esther, I do like the lecture, and will be recorded, and so will be available. This is really great for us. But there is any chance that you can share your screen, get into your site, and show us what it is?
Yes. Oh my gosh, yes. If you've got time, I'm more than happy to do that.
Yeah, it would be great. And I will talk in Portuguese for them, just to ask them to fulfill a formulary, okay, of their opinion about the event. Gente, eu coloquei aqui, eu estou colocando agora no chat, um formulário de opiniões de vocês para dizer como é que está sendo esse evento. Eu peço que vocês preencham para a gente poder melhorar no próximo evento. O próximo evento deve ser com a doutora Kia, também do NC3R, mas vai ser um evento mais específico, porque é sobre como manusear os animais, então eu tenho certeza que o público vai diminuir, mas de qualquer maneira outros eventos virão, então eu peço que vocês preencham para a gente poder melhorar no próximo, tá? Obrigada. Thanks, Esther. It's just because it's faster in Portuguese.
Yes.
Thank you.
Okay, so so you should be able to see my screen. And so this is the EDA homepage. So this is where you come to if you go eda.nc3rs.org.uk. And I do like to point out, if you do want to explain the EDA to anyone and you're not quite sure, we've got this little 1 minute 20-second video on the homepage, and that's a really kind of good description. So if anyone wants to just use that as, this is what the EDA is about, you can give kind of high-level overview. And I'll just sign in. So I've just clicked the sign in button, and I will just sign into the account we use for demos. And this is the part where I hope I've remembered the password. All right, so this is where we come when you first log in. So this is kind of your dashboard to find everything. So if you want a little bit of help, there's some support on the right here. Your most recent experiments are listed here if you've used it before. We've got a flag for when the next demonstration is. It also includes, like, if we're going to do maintenance or update, we'll put some information there. Now, to give you a quick demo, I think what I'll do is I'll start with a template and show you how you can respond to some of the feedback. So we have templates and examples that have different experimental design features. So we've got simple two-group comparison. So that's like the one I used in the slides. If you've got a crossover experiment, here's a crossover experiment represented. We've got one where you as the experimenter don't allocate anything because you are looking at different genotypes and you're kind of seeing, you know, what's happening with the genotypes and things like that. But so you can just flick through, there's 5 examples, and I'm just going to open this one here, the simple true group comparison. And basically it's an experiment that looks like this here, but it's got all of this information that's written out in text here has been put in an EDA diagram. So I'm going to click that link here that says open experiment diagram to view information contained within each node. And this opens our beautiful experiment, and we can go ahead and adjust that. So if because this is a template, I don't own it, so I can't save it. So what I'm going to do is I'm going to save as, and that will give me a copy. So it calls it copy of example 1. And now this copy, I can alter and save and things like that. So this is a mostly built experiment, but not completely. So how we get feedback on our experiments is we go up to the tools in the top here, and we go tools critique. And what this does is this runs our experiment past a set of rules. And these are the things where we got a working group of statisticians and methodologists, and basically came up with if this happens, then we give the person this advice. If this happens, we give the person that advice. And we've got, I think we're up to about 178 different rules. So different circumstances under which we give you different advice. So that makes sure we cover a lot of different cases so we can give you good advice. Okay, so now it's finished. It tells us we have no errors, 3 warnings, and 1 piece of advice. So we can see in the top right here, we have the icons telling us that errors are red, warnings have these little yellow triangles, and advice is blue. So I'm just going to cover a couple of the warnings just in the interest of time. But if we click on this little icon here, it opens this window, and it tells us that our diagram doesn't include any nuisance variables, and it tells us that nuisance variables indicate other sources of variability which may influence the outcome, such as a blocking factor or a covariate. So if we don't have them, we could accidentally not realize that we've got confounding, or we may make our experiment less sensitive. And it goes through and tells us different things that are common in animal experiments. So cages, the room, if animals are not all housed together, the day or the time of the intervention, etc. And then it tells us different ways that we could mitigate against the effects of these. So that can help us decide what will work in our particular experiment. And let's say that in the animal character, so that's kind of the feedback, but the information inside the nodes is in these little, if we click on the little striped icons, we can get extra information. So let's say in this case, we were planning on using male diabetic mice. So we'll just male diabetic mice. And we realized that we should actually use both sexes. And then we realized that we could actually use sex as a nuisance variable. So what we could go ahead and do, so if we use just male mice, we get a warning that says, are you sure you want to use only one sex? So then we can go ahead and add another sex. So because these 2 gray nodes are connected, if I click on the one to the left, I get a little icon that looks like the one to the right. So I can click on that, and that gives me an extra animal characteristics node. So I've got my male diabetic mice. So now I'm going to add my female diabetic mice here. So I'll just say female diabetic mice, and then just say in the sex thing that, excuse me, that they're females. And then what I can do with this nuisance variable is I can say that okay, let's say for our study, we want to know the overall effect of our drug on plasma glucose. We want to include both sexes because we want to make sure our results are generalizable, but we're not specifically interested in the effect of sex. So in our case, sex could be a nuisance variable and we could use it as a blocking factor. So I'll go ahead and add that into our diagram. So if I look on the left here, you can see we've got the symbols for all the different shapes. And if we scroll down, we've got the nuisance variable symbol, and we can just drag and drop those onto the EDA. And that gives us a nuisance variable. And in our case, the nuisance variable is sex, and we're going to add male and female. So we can just add these little skinny icons here. They represent the variable categories, and that is female and male. And then because I want to take the effects of sex into account in my analysis, I want to connect this node up to my analysis node. So to do that, I just grab this arrow icon, I drag it over, we get green corners, that means everything's okay, release, and then we just need to tell this how we're taking account of the nuisance variable. So if it's red, it means it needs more information. So we click on that, we get the spanner icon, we say, okay, that's a blocking factor, and then we can go ahead and get more feedback. So when we go tools, critique the second time, it's taking into account the changes we've made to the diagram. So it's taking into account the fact that we've added the other sex and the fact that we've added a nuisance variable. So it shouldn't be warning us about nuisance variables anymore because it should know that we've got one. And it should be giving us a bit of assistance in using both sexes. So we'll just wait for that to kind of share. And the critique can take a different amount of time depending on how many people are online at once and how many people are clicking it at the same time. So now it's telling us we've got 1 error, 3 warnings, and a couple of bits of advice. And so the error and the warning is on our nuisance variable because we haven't given the system enough information. So errors are about we haven't been clear with the system, or it needs more information in order to be able to give us an analysis suggestion, for example. So in this case, it's saying that if it's a nuisance variable, it has to, and you want to use it as a blocking factor, it absolutely has to be categorical. But it says, okay, if you want to use it as a blocking factor, here's some information. But if you do want to treat it as continuous, say, for example, use it as a covariate, it will give you information about that as well. But in our case, we are happy to use it as a blocking factor, as categorical. So that's fine. So we'll just go inside, select categorical. And where it says how is variable accounted for, we're selecting blocked because it's a blocking factor. So once again, we got into the node by clicking on the stripy icon on the bottom right. So we've actually dealt with, and if we say the allocation of experimental units to variable is, in our case, Mendelian inheritance. So units randomized to categories is something that we can control. Units randomized via Mendelian inheritance is something that, say, nature controls for us. And if we're not randomizing to categories, we just need to say. So, so we've dealt with all the feedback on there. Sorry, I did that rather quickly just because I want to give you an overview of things quickly. And we can see this advice message. This is the message I had in my slides about both sexes being studied in the experiment, helping you take account of sex in the randomization, what to do when you're using it as a blocking factor versus using it as a factor of interest. So that's what we've kind of done with our blocking factor there. And then once we have dealt with all of the errors in the system, we can go tools, analysis suggestion. And this basically looks through our diagram and looks at things like how many groups are in our experiment, what kind of data you're collecting, is it continuous, is it categorical, how many variables are involved, do you have any blocking factors and things. And then it will give us an analysis suggestion based on what is appropriate. And I've got a question about the security of the data and are they downloadable in any way? So with the EDA, as I said before, we do take the security of the system really seriously because people are using this to design experiments. It's, you know, kind of proprietary information. So we do make sure that the system is secure. And in fact, I can't see anyone's experiments in the EDA unless it is specifically shared with me. And also we do have the system tested annually to make sure that we've got good internet security and things like that. But then in terms of downloading, actually, yes, you can download it to either save things to your own computer if you don't want to store anything online, or you can download things and share them and share with other people. So just quickly cover the analysis suggestion. So in this case, the analysis suggestion here is a one-way and over with blocking factor. So it's taken into account, okay, it knows that you've only got two groups and that there's a blocking factor involved, etc. And in terms of downloading, if you wanted to, for example, save this experiment and save it on your computer and then delete it from your account, you can go File, Export, and export diagram data, and that will export a .eda file, and you just go OK, and that will save it to wherever your downloads save on your computer, and then you've got a copy that you can re-upload later. So if you would rather only have it saved offline, you can do that. And so just quickly to finish up, with this diagram, if we wanted to use this to generate a randomization sequence, because I've not connected my blocking factor to the allocation, it won't take that into account. So I just need to do this, use the arrow, and the red link just means there's not enough information. Click on that, call it a blocking factor. And now if I generate the randomization sequence, it will randomize, as I showed in the example in the talk, with group 1 and group 2, and it will randomize the males and females separately. So it will give me separate randomization sequences. So this is how the EDA can take account of things that you want to randomize separately. So that is a very quick whistle-stop tour of the EDA. But as I said, I will be giving a demonstration. And actually, if I go, there we go. So I'll put the link in the chat for registering for a virtual demonstration, because that covers things at a much more reasonable pace. And I encourage you to join me in January or March if you would like to learn more about the EDA. Or actually, if we go to the EDA website, if you want to have a look at a demonstration, but the live demonstration times don't suit you, you can go to this page, and here's a demonstration that I recorded earlier this year. So I'll put that in the chat as well. So you have access to a recording if you'd like to go over the EDA at a bit more of a reasonable pace. So how was that, Helena? Is that a good start to the EDA?
Yeah, it was great. I was thinking about it because sometimes we explain, but we can not see in the site exactly, so it will help us a lot. And it's recorded, so everybody can go back and slowly do something, yeah?
I do recommend looking at the recorded demonstration if you want to get to grips with the EDA, because the tour I showed you just now probably was a bit too quick. And yeah, so I recommend looking at the recorded demonstration where I go through things a bit more logically. So
yeah.
I think that would be more helpful.
Thank you a lot. I don't know if anybody has any questions, because I should let Esther go. And just for instance, Alex, I will let you make the question. I ask everybody, please, you should do the evaluation of the formulary for us, please, so we can get better these events, okay? We intend to do more of these events, and I want to have this getting better and more inclusive and whatever it is, okay? Thank you. Yeah, you can ask Alexander.
I would like to ask how come this brilliant idea of a 3R organization, how this happened? Was a group that united, was a pressure of the society in the UK, was a demand from the journals? How come this happened?
So I think there was a review of animal research in the UK in the early 2000s. And in the end, part of our government, the House of Lords, came up with a report and said, basically, we need to fund an organisation to lead on the 3Rs to make sure that that it gets promoted and that people are thinking about the 3Rs as much as possible. And so in 2004, actually, we've got our 20-year anniversary next year. In 2004, the NC3Rs started. And basically, yeah, it was a report into how the 3Rs are being implemented in the UK led to the recommendation that an organisation was specifically developed and funded to promote the 3Rs throughout the UK. And so, yeah, that was 20 years ago next year. So it's very exciting.
Wow, congratulations.
Thank you.
Great. Thank you, Esther. I will finish the event. I will ask people that are in the meeting room to wait just a little bit. I ask Iber to close the YouTube, yeah? So, Esther, I don't know if you have anything else to say, but we have a lot of thanks to give to you, to thank you for your participation and for everything. You know that you are being very kind